A pleasant afternoon to all. It is my great pleasure to welcome you to the opening of the OMICSena lecture series. Some of you might have attended first OMICSena last March. That was an information field premiere of the OMICSena platform where DOST-FNI featured the frequently asked questions on Nutritional Genomics. For the second time, DOST-FNI brings you OMICSena with a bolder move to bring together omics experts from way beyond the Philippine borders. The Philippines continues to build strong innovative foundations, and learning and reflecting from the global scene is a very way to go. As such, we are thrilled to bring you the 9-day OMICSena lecture series. For 9 afternoons, we are expecting to learn the best of omics technologies and how these emerging platforms can enhance nutrition and food research that will eventually be translated for societal benefits of our country. Beyond basics, hopes are high that we can also build networks and collaborative paths, all towards building an omic-driven, innovative Philippines. William Wordsworth said that life is divided into 3 terms: that which was, which is, and which will be. So let us learn from the past to profit by the present, and from the present to live better in the future. I hope that through all the topics to be discussed from this day until October 7, we can learn how to create a better future in life and nutrition through the studies of omics sciences. To all the participants, again, my warmest welcome, and lend us your active participation because we have inspiring lecture series ahead. So thank you so much and stay. Maraming salamat po at magandang araw muli.
Magandang agham po sa ating lahat. OMICSena was coined by DOST-FNRI to create a scene in a good way. It seems that it will continue to serve as a platform for the DOST-FNRI to keep us in the loop on its undertaking in genomics. With Dr. Brian Gonzalez on board, the second OMICSena for 2021 collaborates with the DOST's Balik Scientist program to drumbeat the advent of another set of omic science at the Institute, the metabolomics. This expansion in the R&D platform will guide DOST-FNRI in setting up more actionable strategies in the molecular food and nutrition arena. I am optimistic that the series will provide new learnings to bring stronger and bolder omics-based research and development initiatives, not only in DOST-FNRI, but in the entire country, as I understand that this OMICSena is well represented by genomics enthusiasts mula Luzon, Visayas, at Mindanao. As we come together for the next 9 afternoons, I hope that we will be able to lay out plans and strategies towards the creation of new intervention systems, biomarker discovery, and innovative health and nutrition platforms through the use of omics technologies. May this event sharpen our vision to acquire the lens of seeing through what our country needs to reach zero hunger and optimum health and nutrition at the molecular level. Congratulations to the organizing committee of the OMICSena lecture series and to all the staff of DOST-FNRI led by Dr. Imelda Angeles Agdepa. Magandang agham sa ating lahat.
A pleasant day to all. The Nutritional Genomics program of the DOST-Food and Nutrition Research Institute is part of the DOST's Big 21 in 2021. This achievement highlights the growing field of nutritional genomics and the DOST-FNRI's New Gen Laboratory, now an ISO 17025 Certified Molecular Laboratory. Beyond this recognition, I am more thrilled to see what our R&D program and scientific and technological services in nutritional genomics can offer to ensure optimum nutrition for everyone. Earlier this year, the first OMICSena answered frequently asked questions on nutrigenomics. The first installment of this web-based learning platform of DOST-FNRI's genomics programs made us realize that nutritional genomics can scale up nutrition and health sciences. Studying our genes through the omics platforms can bring about enhanced nutrition recommendations and perhaps nutrition and health policies in the long run. Its eventual integration in clinical practice can boost our efforts to overcome malnutrition and help us achieve our goals of good health and well-being. As DOST continues to expand the country's omics footprint, this second offering of OMICSena with Dr. Brian Gonzalez, our balik scientist for metabolomics, is anticipated to bring new frontiers in omics-based innovations. This will fine-tune our efforts to use various molecular platforms to pacify and eventually end our long-standing battle against malnutrition and non-communicable diseases. Now, with the generous sharing of information and practices from our keynote speakers ahead of us, I invite you all to join me in getting a detailed glance at omics technologies and how they can play critical roles in nutrition and health. May this deep appreciation of omics technologies guide us in pushing for more innovations directed to our local needs. I am optimistic that this lecture series will also strengthen the competencies of our researchers in initiating omics-based research and development. One thing that the ongoing pandemic has taught us is to hold tight to what science and technology can do. Science and technology will always be an important key in solving various societal problems. Napaghahandaan man o hindi, katulad nitong pandemya, innovations and discoveries will help us thrive and become resilient. On behalf of the Department of Science and Technology, I would like to thank our speakers from different parts of the European Union and the United Kingdom for their time and expertise. I extend my appreciation especially to our DOST Balik scientist, Dr. Gerard Brian L. Gonzalez, for bringing this exciting series to light, together with the DOST Food and Nutrition Research Institute. Thank you all for being here. Let us have a learning-filled week ahead. Mabuhay!
Good afternoon, Pilipinas, and good morning, the Netherlands. Our sincere apologies for the technical glitch earlier. It is a gloomy afternoon here in Manila, but let us keep our senses awake this first Monday of October. I am Jay Cosnasis, and welcome to the second week of the OMICSena lecture series. Last week's lectures are uploaded on the official YouTube page of DOST-FNRI, and if you want to watch them on demand, please feel free to do so para OMICSena all you want. We are bound to witness lecture number 6 this afternoon with another esteemed expert. Before I give the floor to Dr. Gerard Brian Gonzalez, our DOST Balik scientist for metabolomics, may I again inform everyone that DOST-FNRI will celebrate its 75th anniversary in July 2022. The OMICSena lecture series is part of its countdown, a unique way of living through its promise of being your kasama sa bawat yugto ng buhay at kabuhayan. To visually start another learning field episode of OMICSena, please welcome our balik scientist for metabolomics, Dr. Gerard Brian Gonzalez. Over to you, Brian.
Maraming salamat, Jacobs, at magandang hapon po sa inyong lahat. First, I would like to thank our secretary, undersecretary, and the director of the FNRI for the warm welcome. And to all of you who have been actively participating last week and also today, we have a special treat for you again. I would like to introduce to you a colleague of mine from Wageningen University, Dr. Justin van der Hoof. Justin is an assistant professor in computational metabolomics at Wageningen University, and he develops tools to enable biochemical interpretation of spectral data obtained from complex metabolite mixtures through structural and functional annotations. He is developing computational metabolomics approaches inspired by two fields, such as natural language processing and genomics, and he applies these technologies for plant root microbiome and human food metabolome studies. So without further ado, I'd like to give the floor to my colleague and friend, Dr. Justin van der Hoof. Justin, the podium is yours.
Well, thank you so much, Brian, for this very kind introduction. And thanks everyone for joining. Let me share my screen. Can everybody see it? Okay.
Yes.
Okay, thank you. So, well, thanks for the introduction. As Brian said, I'm a lecturer at the Fragments group in Wageningen. And over here, it's also a gray morning, but they promised us some sun for today. So let's see. So for today, I was asked, could you tell us something about metabolomics, annotation? And because I think that many of you may maybe starters in the field, I decided to start at the beginning, and then we see where we can end. By no means I want to finish my slides. If you have any questions or want to know something about what I tell, just type a question in the chat, and Brian, maybe you can have a look, and when there's a question, see at which moment we can deal with it. So I prefer that we can discuss any unclarities during the story, so at least we all understand where we're going. Okay? Cool.
Yes, that's clear.
So, and then before I start the story, I would just like to highlight that many of the things that I tell today are definitely not done by me alone. I think metabolomics as a research is definitely a multidisciplinary field, and therefore I would like to thank all my former colleagues and current colleagues for being great in terms of teamwork and collaborative efforts to develop the tools and to apply them to interesting fields of science, including plant metabolomics, nutritional metabolomics, and bacterial metabolomics. And yeah, so this is a word cloud of I think of 2 years ago from my LinkedIn page, and this kind of provides you with some sort of idea of my background and expertise. And yeah, maybe unsurprisingly, we see that metabolomics is appearing a lot in the abstracts of the papers I publish, and data, molecules. But you can also see that genomics pops up there, and some of the applications that I've been working on, like lifestyles, microbial, and a couple of other things, plants, for example, over here. So that already gives you actually quite a good idea of my background. So I did my PhD in Wageningen, where I worked on plants, tomato and tea, and identification of the compounds they're in, using LCMS, so liquid chromatography coupled to mass spectrometry, and also NMR, nuclear magnetic resonance spectroscopy. And then I moved to Glasgow, where I did several postdoctoral contracts, and that those included bioavailability of flavonoids. So, for example, they are highly abundant in tea and chocolate, and I investigated how they are being processed by the body and by the microbes in our gut, and how they end up in our urine. And then now I'm back in Wageningen, and over the last years, I've been also investing time in how can we combine genomic workflows, in particular genome mining for interesting natural products that for which the genes are encoded in the genomes of organisms. How can we link those kind of workflows to metabolomics workflows that I have been working on as well? So that brings me to a typical metabolomics workflow, as you may have seen it already during these lectures. And that is where you take a sample series, for example, here of strawberries. You generate profiles. It could be either through LCMS or GCMS or gas chromatography coupled to mass spectrometry. You get chromatograms. You do a lot of pre-processing. You start to analyze the data using statistics, and then you do the identification of selected metabolites, right? So, and that is then your data interpretation, where you want to couple annotations or identifications or structural information to the chromatograms and the signals that you obtained. Otherwise, it's very hard to do any biochemical interpretation. And actually, that already represents one of the key challenges in the metabolomics field. I don't know how many of you actually already have some experience with metabolomics, but I know that Brian has, and I think he will. I hope he will agree that to annotate the structures to these peaks and signals, that is a key challenge for the field, especially in complex mixtures. So I will show you some examples now of how we tackle that, and how you could also make use of the various information that you already obtained if you do an LCMS. So I will mainly focus on mass spectrometry today. But if some of you are highly interested in MR, I can also show some slides on that. So I think that I also leave that a bit to the audience. So yeah, I will discuss some chromatography. I will definitely discuss some mass spectrometry fragmentation, because that is also a cornerstone of my work, and then show you how we can use that for annotation. And then if time is there, I can also introduce the concept of molecular networking and library matching, okay? And let you actually practice it a little bit with that. So why is it so hard to do metabolite identification? So because you would argue, we know already quite a lot of the things that are in natural samples, and we get signals. So isn't it just a matter of looking at the databases, what is in your samples? Well, in some cases, but in many cases, the diversity of the chemical structures in the complex mixtures really hampers that process. So there's many different structures possible. You just get many signals, and I'm aware that for some of you, this terminology is maybe a bit vague and abstract still, but you can keep in mind that you have many metabolites. So each metabolite produces at least one signal. And then we have also a lot of so-called ion products. And that, for example, there's an example here on the slide. So we see this is the monoisotopic mass. And then we actually see that already has 3 additional mass peaks associated with it for free. But we have to deal with them. And this is due to the natural abundance of so-called isotopes of atoms. And we also have adducts. I can explain those easily. So for example, with salts, if there's salts in your sample, they often glue to your ions in the mass spectrometer, and you can also see them as a peak appearing in your spectrum. And the final one that could also add to the number of ions is in-source fragments. So that is also easy to visualize. You imagine the metabolite is fragile in the gas phase, and it simply breaks apart when it, for example, is treated inside the mass spec to guide it towards the detector. So that also can lead to additional ions. And then finally, we have simply a lot of unknown unknowns, right? And if we look at urine, which is an interesting sample matrix, then we know the main constituent, right? That's water. And we also actually know of the remaining things, the main constituent, that's urea. However, then it becomes really a challenge, yeah? And that because the contents are really dependent on what you ate and drank yesterday, for example. the classical route is that, which I explained, yeah, in the typical metabolomics workflow, is that you first start to identify using statistics the interesting features, and then we will try to identify them. So here we see a comparison of of a beer, particular ale from Scotland, versus a pooled sample of all the beers. So then I'm able to pull out the interesting features that are more abundant in this particular ale. And on the right-hand side, you see an example of how you can buy standards in the shop, in this case, Sigma. And is there a question? In this case, in the shop. And then you can start to compare those signals, right? And if we zoom out a little bit, so what are the critical steps to get from a feature? Go back, so feature here, a dot over here, yeah, to a molecule, right? So you typically start with, let's say, a rotation time in your chromatogram and a mass, right? And you know in which mode you obtained it. So I'm assuming you got some background in how a mass spectrometer works, but maybe that's not the case. However, so typically you can either run it in positive ionization mode or in negative ionization mode. And then we can start to match your peak to a reference compound. And that is usually the first thing then that you try. However, you have to realize that in many cases, there won't be a match to a reference compound. So the second step then is to find the molecular ion. And remember, with the isotopes and the in-source fragments, this can be a challenge already, and get the most likely elemental formula. So continuing, we can do a spectral matching if we run fragmentation, and I will show some examples. We can do spectral matching to experimental or in silico MS/MS data. And then finally, we can do some compound classification. So for example, using a retention time or a mass spec. And also there, I will get to some examples. And the final thing is usually that we want to confirm the annotation using a newly acquired standard or additional spectral data, for example, from NMR. And then, first of all, retention time information, right? So I will focus on liquid chromatography today, but you could also do GCMS, okay? So, and typically we get retention times, and that is the measure of time it takes to elute from the column, okay? And that is representative of the interaction of the molecule, of the metabolite, in the mobile phase and the stationary phase. In other words, the metabolite is bound to the stationary phase for a certain time, and depending on the solvents that pass, it will at some point, dissolve, or the interactions will weaken, and then it will travel along with the solvent out of the column. Okay. And then that brings me to the first step that I mentioned. So the use of reference compounds, of course. If this is your chromatogram in orange and in blue it's your standard, then you can happily say, okay, I see a correspondence in the reference compound and the chromatogram, and I can annotate or identify the molecule that is in my sample. Okay, so that is really dependent on co-elution with reference compounds, and you want to try to get independent properties, for example, mass and retention time, to make the identification. I'm just going to ask, is this concept clear to all of you? Could at least some of you indicate that in the chat?
Yeah, they've had a 1-week session last week about some of these principles.
Okay, cool. So actually, that's good then. And they see it again, and I hope that will further solidify their knowledge. And when I worked at Glasgow Polyomics, we had a set of nearly 180 standards that we routinely matched to samples. And I just want to highlight that really it depends on the choice of platform, what you can see. Over there, our main platform was Helic based, so hydrophilic chromatography, and that means that you're good in detecting pure metabolism, energy metabolism, TCA cycle things, but you're not so good in seeing the lipid and the stirred metabolism, because those molecules do not retain very well at the Helic column. So still, if we continue, then you may be very lucky if 30 of reference molecules match in your sample. And you're even more lucky if some of them are actually in your significantly changed metabolites, right? So we really need additional information to annotate the other stuff in this picture. And I realize that some of you may have seen this iceberg before, but I think it's a really good metaphor of what typical metabolomics workflows still see, right? Only the top of the iceberg. And when we continue, then we can also use how you call it? We can use the information from retention times to actually classify molecules. And this is something that you may or may have not seen before, but for example, on an C18 column, we can identify which class of molecules elute where. And you can see examples of chemical compound classes on the slide. And apologies, this is on a Helic column, and this is on a C30 column. So that is a non-Helic column. And what we can see over here is how the various xanthine derivatives are eluting in a particular order. And you can also connect them to the samples, in this case, food samples that you typically can find in. Okay. So again, we can use this for annotation and identification already on the base of classification. Even peak shapes you can use, right? And you can also, or you should use them to make sure that your method works well. Yeah. Remember that for any approach you use for annotation, it will really also depend on good quality input data. Following the principle rubbish in is rubbish out, right? So you want to prevent the rubbish. Look out for good peak shapes. But in some cases, and that is what I wanted to say here, is that some molecules simply do not want to behave well. And you can also use that to annotate structural information. So we want to assign molecular formulas, right? I mentioned that as a key step. And that works then as follows. You typically get a mass. In this case, let's say 46, and then you look at which combination of atoms are possible to fit in. And over here, you see how a lot of different combinations are possible, but only some are actually realistic. That is my key message here. So NO2, CH2O2, and C2H6O, those we know, right? So ethanol is a very famous or infamous molecule, but also formic acid, I think many of you will have heard of. Of course, we can see the downside of having non-accurate masses here. The more imprecise we know the mass, the more different options are in theory possible. And of course, we can use some tricks like the nitrogen rule and the double bond equivalent to kind of see if if a formula is realistic or not. I will give you an example. The double bond equivalent is based on the atoms, the number of double bonds that you can expect. And if that goes to -6, then you know that something is wrong, right? So that is, I mean, maybe in theory it's possible, but in practice it is an invalid elemental formula. So I already mentioned the adducts and the fragments. Here you can see an example of how many of them you may or may not expect in your sample. Of course, this is sample dependent and matrix dependent, but still it is good to keep in mind that only a minority of the signals is actually a molecular ion, both in positive and in negative ionization mode. In positive ionization mode, we see, for example, that the salt adducts are pretty abundant. Over here, we see also the chloride adduct that is pretty abundant in negative mode. So again, mass spectrometry, one reason why we see so many signals is because each metabolite causes multiple ions to occur in your spectra. So, as I said, one of the cornerstones of my work to improve our annotation capacity is mass spectrometry fragmentation. So typically, when we look at the mass spec as this gray box over here in the middle, then it has to do several things, right? It has a source where ions arrive, so molecules, metabolites that are in the gas phase and charged. In this case, you see with positive charged. Then, of course, in the mass spec, we need to attract them with a negative charge, and then we need to do three things, right? Separation of the masses. We need to trap them, and we need to detect them. Yeah? So, and the trapping is typically done, well, in order to detect them, but also we can trap them to fragment them, and that is what happens here, right? So we select this mass over here. We add energy, typically in the form of gas, inert gas like argon or helium, and then then the fractal ion breaks, and then we get a fragmentation spectrum, and that is typically then the fragments, and we can use them to puzzle back what the structure of the molecule was. And we have different options these days, right? We can compare the spectrum to experimental data in public libraries. We can do in silico annotation using the fragmentation spectrum, or we can follow computation workflows to do metabolome mining, like molecular networking. So, interestingly, and this is also something you will have to keep in mind, is that when you do fragmentation, it will not give you always the same fragmentation spectra for the molecules. It is really dependent on many factors, and one of them is actually the type of instrument you use, and the other one is the energy you add, so the amount of energy. And let me show you an example. This is an instrument from Thermo, where we have two places where we can fragment molecules, over here in the ion trap part, and over here in the orbit trap part. And when we fragment this particular molecule in the ion trap part, we get this. And when we fragment this particular molecule in the orbit trap part, we get this. And you already see that these two spectra are different, right? But they also share something. The fragment 60, and the fragment 103, and 102. So even when one molecule fragmented in two different places of the same instrument already yield a different fragmentation spectrum, and this already also shows you that yes, it can help us, but we have to be careful and we have to understand what's going on. So histidine is my favorite victim in terms of fragmentation. And I also would like to add that in many cases you will see in literature that it's a barcode or fingerprint, the fragmentation spectrum, and that really comes from the fact that in many cases it represents a kind of unique way of telling about the metabolite. And what we see here is the molecule histidine, and it consists mainly of two parts, right? The amino group part, and then the side chain, the imidazole ring, okay? And we see over here the mass fragments, okay? So over here, and then we can also see a neutral loss. And what is a neutral loss? Well, that's the difference between the molecular ion over here and the fragment. So here, over here, the difference is 46. And then the question is, in some cases, we can really assign that to a part of the molecule. So we can puzzle where, how can we fit in the fragments in histidine, and where can we find the loss? In this case, we know the structure. So let's have a look. And when we look at these fragments, they contain two nitrogens. And over here, in this area, we see that there are two nitrogens close together. So I think it's a good hypothesis to say that many of these fragments are derived from this imidazole part. Over here, the difference is 46, and we can see that it's 1 carbon, 2 hydrogens, and 2 oxygens that are different. And now we can clearly see that this is due to the loss of this carboxylic acid group. And that is nice, because now if other molecules show the same neutral loss, we can already start to hypothesize that they also have this carboxylic acid group as a substructure. And actually, the things that we just discussed on this slide are the basis for most of the computational workflows that you will see these days. Yeah. So let's recap this. So we can look at fragments, and if we start to see that they are derived from a particular part of the molecule, or in other words, we can make a connection between substructures or scaffolds and the fragments. Then if we see them in other molecules, we can infer that they also have that structure, that substructure. Okay, and the same is true for neutral losses. So I hope this concept is clear. And now let's look at the different energies. I told you that's another important factor. And let's slowly ramp up the number of the amount of energy which we fragment my favorite victim. And then we see that the lower energy, we see only 1 fragment. And we see the neutral loss that we already assigned to a carboxylic acid group. But we don't know anything else now about the molecule. And when we ramp up the amount of energy, we see that, hey, the first fragments start to arrive. That also tell us something about the imidazole ring. Yeah, I told you. And then when we further ramp up the energy, we see that we see both. We see both the loss, and at some point we see that the parent ion is gone. So then it is harder to assign the correct neutral loss again. So you can see that for this particular metabolite, we are best off with an intermediate amount of energy, but this will be different for each metabolite. And this is an example from a paper published 6 years ago now, and that's an azocarnitine that I fragmented. And again, if we ramp up the energy, we'll see how the spectrum changes. Okay, so first we can see the typical neutral loss and fragment for azocarnitines. And if you're interested in this particular group of molecules, I refer you to the paper. But if we ramp up the energy, look at this, right? Same molecule, just a tiny bit more energy, and the spectrum completely changes. We see different peaks now, and if we further ramp it up, we see again how it changes. Yeah, and in this case, this different energies could help me to assign the very plausible substructure of dihydrofolinic acid that is then connected to the carnitine backbone for this azocarnitine. So a takeaway message from this just short series of slides on the energy is that using different energies can help to get more structured information about your molecules, and also that if you can, you could say use the different energies for the same molecule or combine them into one consensus spectrum, using, for example, stepped collision energy on Orbitrap instruments. Now we have all those spectra, and say that you manage to get good quality spectra, then we want to identify them. Say that you don't know what the molecule is, and now we want to match the spectrum to libraries. And one example is MZ-Cloud. You also have MassBank, GenePS public libraries, and Matlin as examples of where you could go and compare your data to. So over here, let's say we match our histidine spectrum to MZ-Cloud. Then we get as best match carnosine, and then the second best match is histidine. And histidine was actually the molecule that we fragmented, remember? So that is good. And you see, actually, if we look in more detail, that all the molecules in the top hits are related to histidine. So also carnosine. So that is good to know, and that means that if known structures with known spectra are present in libraries, and we have the right spectra, then we can expect some good matches. And then we move on. So, and using tools like Magma that was developed by Lars Ridder when he worked at Wageningen University, you can also, that can help you to do in silico analysis. And in this case, again, I used Histidine because now you can see how the program automatically assigns plausible structural annotations for your fragments over here. And we already discussed them. So it's easy for us now to confirm that indeed these are imidazole related fragments, and Magma also assigns them as such and gives Histidine as the top hit, the best hit based on the silico fragmentation. So Magma presents you with a number of candidate metabolites found in databases, structured databases, based on, for example, the parent mass and a certain deviation of that. And it can then help you to annotate the spectra in the substructures, and also shows you the MS data. We can also apply this to carnitines, and that's what I showed in the paper I previously put on the slide. So I managed to annotate very quickly quite a few azocarnitin species using Magma. So again, we have the same setup, but here now we see how it recognizes different substructures. Over here, the 144 is actually the carnitine substructure in azocarnitins. And when we zoom in, we can actually see that more clearly, and also how 85 is indeed a fragment of the azocarnitine molecule. And over here, we can also start to see fragments of the acyl chain of this particular azocarnitine. And then 10 to 15 species were annotated very quickly. I think with nowadays instrumentation, this number can be easily much higher. So in order to assess or establish the right annotation workflow, you have to ask yourself a number of questions, right? And that the first one is maybe, do you know a lot about your sample, right? About its contents, the complexity, and the origin. And if the answer is yes, you can ask, do you know how many expected metabolites of importance are for your study? And if the answer is yes, then I would definitely use targeted MSMS-based methods. Why? Because the analysis workflow is very straightforward, right? You only select those things that you are interested in, that you know should be there if a certain treatment is done, and then you can easily do the statistics afterwards. If the answer here is no, but so you don't know many expected metabolites of importance for you, then you could say the same thing, but now about compound classes. If the answer is then yes, you can use still targeted workflows, MSMS-based workflows, where you don't target specific molecules, but compound classes, for example, based on retention time and presence absence of certain masses or neutral losses. Again, because the data analysis is very much more straightforward. If the answer is no, then of course, we will need to use untargeted MSMS acquisition methods, where we can still use targeted data processing methods, right? For example, to fish out the specific parts of the chromatogram or masses based on, again, diagnostic ions. So keep in mind that I think nowadays instruments are becoming more and more better. So actually, you can also decide to anyway run untargeted MSMS acquisition and then do the selection in the processing step. So you can always go back to your untargeted data if you come up with a new hypothesis and test it on the untargeted data. So, of course, when you don't know a lot about your sample, for example, simply because you haven't controlled a lot of things, let's say urine, and you haven't controlled a lot of things in terms of diet and medicines, then for sure, the best thing to do is to do everything untargeted, and only afterwards start to apply semi-targeted or targeted processing methods. Alternatively, you can of course do metabolome mining strategies that are completely untargeted. And one reason why you would do that is that because the number of MS/MS spectra in our samples is really increasing because the quality of the instrument is increasing. And you here see an example of a file that I run in Glasgow, and each diamond is a fragmentation event. And over here we have the mass, and over here we have its retention time. So a number of things to say about this picture. And the first thing is, yes, we see that we can cover quite a range of masses and also along complete retention time area. But we also see the need for improvement of acquisition methods. Why? Yeah, look at here. Look at the broad band of diamonds that around a mass of 100. Those are clearly not real molecules. In many cases, we are also fragmenting noise. And of course, there are ways to get rid of that in your processing and pre-processing. But together with Glasgow, we are also currently investigating ways on how we can do this data acquisition in a smarter way and ignore the noise and actually get more MS/MS spectra of real metabolites. Okay. So, and one thing that you can do, and that is also actually kind of finding a sort of a new wave of interest in literature, is to apply filters on your mass spectra, MS/MS data. And again, I will use the acylcarnitine example, but you can also design your own filter for flavonoids, and other food-related molecules. And over here, I designed this accurate mass filter of the mass fragment of C4H5O2 and the neutral loss of C3H9N. And you can see over here on the top, that's all the spectra where we see this 85 fragment. And on the bottom here, we see the neutral loss, okay? You can also see that in some areas, the neutral loss is present, but not the fragment. So we can already see that in this area of the chromatogram, they're both corresponding, okay? And basically, I basically could use that to really quickly extract acylcarnitine species, and I've been able to annotate a lot of species that were not previously reported back then in a database like a human metabolite database. So I hope I showed you with this set of slides how there are different ways to get from a feature where we only know the retention time and the mass to a specific molecule. And over here on the right-hand side, I show that for a metal histidine, where we go from the feature to a level 3 annotation, where we can classify the metabolite. For example, here we know it's an amino acid-related molecule. And so you can slowly get some ideas on the affected pathways. Then level 2 is a database comparison. Maybe we can then see that it is a metal histidine, but we don't know where the metal group is. And then we need to still do some targeted screening or NMR or something else to assign where the metal group is connected to the histidine. Okay, and these levels, these 4 identification levels, are derived from the Sumner et al. Paper back in 2007. In the meantime, there's a couple of other classification schemes for reporting identification levels in metabolomics appeared in the literature. One of them by Emma Szymanski et al. That is really also fully devoted to mass spectrometry. So I will use the remainder of my time to introduce some aspects of metabolome mining, and then I would like to have some time for questions as well. So I will switch the presentation now, and then that will allow me to introduce the concept of metabolome mining. In the meantime, Brian, have a look if there's any questions. I'm happy to answer them on this part.
I don't see any pressing questions. Jacobs, do we have anything on YouTube?
No,
I think so
no
far. I
questions.
think so far it's clear. I hope, at least.
I hope too. That's great to hear. It's always very hard to when you do this online lectures to to assess whether the audience also thinks it's clear. So yeah, really feel free also to put in the chat that it's clear or that you can follow. That gives me, is it going? Apologies. Let me, okay. My bad. Okay, I hope you see a black screen with some colors.
We do.
Is that correct?
Yes, we do.
So, and that is just to show you again the concept of fragmentation. So we have a bunch of masses of different size, and in the mass spec they're typically concentrated and then separated, for example, based on the time of flight or on the frequency of the orbit in the orbit trap. And then we can also assess the relative abundance, based on the number of ions. And it's important to keep in mind that mass spectrometry is a really strange technique in that sense, that there's all sorts of things that influence its abundance, right? The matrix, and also how well an ion actually is able to attract and retain charge. The second step of importance for metabolome mining is the fragmentation. So there we select an ion and then we fragment it, and the hammer is typically inert gas, and then we get the fragmentation spectrum. And in literature you will see MS2, you will see MS, and you will also see tandem mass spectrum. In my opinion, those are all almost equivalent terms. So don't feel lost when you see all this terminology. It's just important to know whether we discuss the MS1, the full scan data, or the MS scan. It's also good to keep in mind that in some cases people have managed to continue to MS3 and MS4, and that simply means that you select another here a fragment peak, and you then do again fragmentation on that. So here we really get to the empowering step using metabolome mining. And I showed you this typical workflow already, remember? And the key thing now is to keep in mind that we don't do this selection, right? We used all the data, and that has a reason. So we don't do first the data analysis, the statistical things, and select our ions of interest. No, we take everything in. And I know that I showed you already quite a lot of pictures of spectra, etc. But when I was starting in this field, it really helped me to start to understand how the data was obtained and what kind of data structure I had in order to be able to actually annotate the molecules. Back then, first by hand, and later on using computational tools. And over here, I just wanted to highlight how in an orbit trap situation, we have a duty cycle. And so every cycle, we have a full scan, and then we have a number of MS scans or MS2 scans of typically the most abundant ions in your full scan. And in that way, we can fragment usually quite a lot of masses these days in your data. And when we do extracted ion chromatograms, you can see how at several places peaks occur, and that in hopefully quite a lot of them can be selected for fragmentation. Also, when we are able to combine information from full scan data, so also the abundance of the LCMS peaks, together with MS information, we are able to both do a qualitative analysis and a quantitative analysis, and really extract powerful information from our samples. So today there won't be time to practice yourself, but there are on the internet already, for example, the Dorstein YouTube channel. There is plenty of workshops already available where you can follow the steps that are needed to do parts of the analysis. In that case, in the GMPS environment, of course, for example, to do also some statistical analysis using the GMPS dashboard. Well, typically, this data-dependent acquisition workflows, they result in thousands of spectra, right? MSMS spectra. Even in one sample, you can get more than 1,000 MSMS spectra, let alone if you have 50 files in your experiment. So 50 times 1,000 is 50,000 spectra. So, of course, if you have the right knowledge of the mass spec, you can try to decipher how molecules and fragments are related to each other and assign IDs to the various fragments in your data. You see an example over here, right, of how we slowly go from a large mass down to smaller masses. And you see how Annelies and colleagues did a very good job. But of course, in some cases, it is very hard to do this, and you really need a lot of background. And now I come back to my favorite example of why we need mining. So all this diamonds, all this spectra, if we don't do the selection, we need to find another way to organize and structure the data. And molecular networking and substructure finding are really, I think, very nice ways that are increasingly being used to do that. Over on the right-hand side, you see an example of a molecular network, and each dot is a group of highly similar spectra, and and they are connected if they're spectrally similar to each other. And the color in this case is based on the chemical compound class. So for today, I cannot go into detail of all the different tools that are already available from GMPS. I will really focus on the basics of molecular networking. But I can really encourage you to if you are starting to use LCMS-based metabolomics, and especially in the food field and also bioavailability field, and you're planning to do untargeted metabolomics, do read some of the GMPS based tools, papers, and also other related software tools to get an idea of what is possible. So you can actually ask the right questions, because if you know what tools are available and you can run, you can also adapt your questions in such a way that you can fully exploit those tools. And take a moment to look at this picture. And then if some of you can at least put in the chat, tell me what you see, and don't take too long. Okay, I see some answers. Okay. So some of you see only one aspect, and others see there's a separation between seals and birds, and that is exactly why I was taking this picture. Right, I took it myself near San Diego, and indeed, our human brain is very good in recognizing patterns. Yeah, maybe you didn't know or realize, but it's really good, especially when it comes to subjects like animals, and it's also really helpful. We can separate a lion from a cat, and we can separate an elephant from a giraffe, and we can assess its danger. Right, basically that is the reason why. And over here we also see the two groups, and actually that is the principle of molecular networking. So you want to find the patterns in the data and structure it, right? And you can then exploit that, right? We can exploit it to propagate structural features. So if we know that the seal has two brown eyes, I don't know if that's true, but then we can hypothesize that all of them have two brown eyes. And we can also annotate family chemistries. We can, if we know the compound class of one molecule in a family, then we could infer that all of them are of that family. And the reason why we can do that is that metabolites share biochemical substructures. So like Lego building, we can take common substructures in your data, like in food, in plant-based data, it could be flavonoids, quercetin, or a glucose molecule, and we can combine them and build our molecules. And the nice thing is that in many cases, these things are revealed in fragmentation spectra as substructures often produce similar fragments or neutral losses. And we discussed that for histidine before, right? However, we don't get the annotations with the spectra usually, because we typically do untargeted metabolomics when it comes to complex samples. So the first port of call is really library matching, and and the nice thing is you can do this these days within a lot of the tools. And this is an example that I show you here more schematically. We have MSMS data. We compare all of them through library spectra, and this could be from GPS and all the others that I mentioned before. And then we get a spectral match where there's the reference molecule and your experimental data, and this is typically called a mirror plot that can help you to assess the validity and the reliability of your match. But Justin, that's all well, yeah, but should I do that for each comparison? No. What we also did is we designed some scores, right? And the basis of library matching and also molecular networking and of all sorts of other things is a mass spectral similarity. And that is what that is the last complicated thing that I want to share with you. So forget about the formula, but just focus on the three spectra on the left-hand side bottom. And then by eye, for yourself, assess which one are most similar. Yeah. Okay, I think you already could have seen, and let's see if you have it right for yourself. If we would put those spectrum in a kind of multidimensional space, then typically the red and the green, and I do apologize, I should change these colors at some point, but the top spectrum and the left-hand side spectrum are most close together, yeah? And they are also most close together in this multidimensional space. Why? Because they share most fragments. And then we can simply calculate the angle in this multidimensional space and also assess the other angle. And if we then take the cosine of those angles, and for those of you that did their maths at high school, you will know that when the angle is really small, the cosine is really high. So that means that if two spectrum are very similar, they end up in a similar position in the space, and their cosine angle is almost 1. And that's neat, right? So if the score is high, if it's 1, then the spectra are identical, and if the score gets lower, then they're less identical. And that is the basis of mass spectral similarity scoring, and that is the basis of library matching and of molecular networking. So it's also important to realize it's really also based on mass fragmental overlap. So if you don't have any overlap in fragments, your score will be 0. And if there's overlap of abundant fragments, then the score will go up. So, and typically we can validate our matches, and we have 2 examples here on the slide. Using a mirror plot, we can look at the parent mass differences, we can look at the number of ions that match, we can look at retention time information if we have, but also at metadata. And you can use your own common sense and eyes. And that I wanted to show here, where you have 2 examples. And I hope you agree with me that the top one looks to be a better match than the bottom one, because here on the bottom hand side, you see that there are 2, particularly one really abundant fragment that is not found in the top spectrum. But and over here, we can see at least a partial match, right? we see that this fragment here is not matched in the mirror plot, but all the others are, and they also have the same intensity ratios. So that is a really good sign that we're onto something, and at least we can use partial information from our library. And that is also how I assess library matches. So I will skip this part in view of time, but be aware that every now and then there's tutorials organized by the GPS team or by satellites in Europe and in other places where you can actually also practice this for a bit and then discuss together the results with the moderator. So, and if you're interested in this kind of topics, then I can really encourage you to do so. So to finish off for today, some words about molecular networking. And one thing to also realize there is, I told you already about similarity scoring. And the assumption is that similar molecules fragment in a similar way, that spectral similarity equals structural similarity. And please be aware that this is not always the case. So mass spectrometers are strange things in that sense that we cannot still understand completely how they work. We only know that if you throw in the same molecule, the same instrument, with the same conditions, you typically get the same spectrum back. But the full understanding of how the process works is still not, still not fully understood. So in that sense, we can only work with what we have, and in many cases, this assumption holds, but not in all cases. So we need this comparison, and we assign a score, and we can use the score as a threshold to network the molecules, right? That is the beauty of scores. You can do that at a large scale automatically, and you can say, okay, if the spectral similarity score is above 0.7, we break the edge. We draw the edge, and below we don't draw the edge. And that is then the basis of molecular networking. And remember the seals and the birds? Well, basically, we're going to do the same thing over here with the 7 spectra. But if I ask you to do it by eye, you'll probably realize it's a much harder task. And that's because in human evolution, we are obviously not trained to do this for spectra. But luckily, we designed algorithms that can do it for us. So the algorithm can recognize which spectra belong together, and it can assign the scores. And you can see how we can draw edges here and on top and on the right-hand side, and no edges between these 3 groups. And then when we would zoom out, this could be part of a larger molecular network. And that is here drawn on the right-hand side. It's of urine samples. And we can then again use the library matching that we just discussed to do some seed node annotations, in other words, to start to annotate this network. And over here, we can see how terpenoids, flavonoids, and other things are being distributed over the network. And then you can start to assess and assign structures to your entire family. And I will give you 2 examples. So first, the contaminants over here. And that is, if we see multiple matches to contaminants, then we could safely assume that that is a family that is of no interest to us, and we can ignore it for further analysis. Over here we see the terpenoids, and they could be of interest. And if this would be urine samples, then we could say, hey, maybe this is a family that is derived from molecules from plant-based food. And the same is true for the flavonoids, and also pharmaceuticals in urine are more common. They can be derived from drugs that are being taken. Okay, with this slide, I would like to finish this part of this presentation, and also thank you for your attention. And let's see if there's any questions.
Thank you very much, Justin, and also thank you for reminding our participants on the difficulties and also the challenges, but also the fun of doing metabolite annotation and LCMS in general. So to remind everybody, even on our first day with Professor Lars Drakstedt, he already mentioned to us that metabolite annotation remains the bottleneck of metabolomics. Nowadays, you can easily with high throughput machines analyze hundreds of samples in a few weeks, but then data analysis of that will take easily double or triple the time it takes. And we're very thankful for people like Justin for developing the tools that we need to help us identify. There are a few questions I'd like to raise to you, Justin, from our audience. First, from Dr. Cation, what happens? How useful are those ions then that are lost due to ionization? Can we make use of them? I think what she meant was ion suppression, or maybe from the fragmentation in source fragments.
So if it comes to ion suppression, I think that is what I mentioned, right? So, well, if you see that, then I think that's a sign that you have to think about if you're interested in specific metabolites in that area, that you may want to, for example, dilute your sample a bit in order to allow for more different metabolites to pick up the charges. So it's not so useful, right? I mean, ion suppression, it is not a useful thing, but it happens when the mass spectrometer gets saturated with ions. And if you talk about in-source fragments, of course you can use them, right? You can in strategies like all ion fragmentation, they are actually being used, right? So, however, there is an additional challenge. Because in that case, you have to puzzle together which fragments belong to which parent mass. And there's programs like MS-DIAL and others that can help you with that, but it is a real difficult puzzle. So you have to have some experience in order to assess the quality of your so-called deconvoluted spectra, and then you can compare them to libraries, etc., etc.
On a personal note, I'm quite curious how accurate the data of the mass do you require for the fragmentation? I mean, would you need then a really high resolution, or a QTOF spectrum would be sufficient?
It's a very good question. So, and I would refer back a bit to the scheme that I try to explain, right? So, do you know a lot about your samples? If the answer is no, then I would argue that accurate mass, like from an Orbitrap or good TOF, is needed to do good annotation, right? If the answer is yes, then you may more easily get away with ion trap data, even because you can already know what to expect, right? You can prepare, you can use the right columns and methods to kind of steer that process away from the things that are interference and really much more targeting the interesting things.
Okay, for our next question, I'd like to ask, you have a fan mail here on the chat box, Justin. So if I could ask Ralph Molino to unmute and ask your question and give your comment yourself.
Hello, sir. I'm Ralph from the University of the Philippines, Diliman, under Dr. Hunya's group. So I'm a fan of your work. Actually, I use GNPS and MS2LDA on a weekly, if not daily basis. So my first question goes beyond your presentation, but more of as a user, if that's all right, sir.
Yeah, go ahead.
So, sir, I have used a number of in silico fragmentation softwares, such as MetFrag, the Replicator Plus, and so on. I usually have an idea on the compounds, but most of the time, these softwares give different candidate structures. Many of the product ions they show are weird in the sense that they will be difficult to produce based on known gas phase reactions. Sir, will you agree that there are possible room for improvement on these many softwares, and how do you think they can address their limitations? That's my first question, sir.
Yeah, so that's a very good question. And then, if I would translate it so I can answer it for the entire audience, is that in silico tools can often provide different answers, right, for one spectrum, and sometimes the answer looks very strange. So how can we best use the information? Yeah, and should we trust actually what the tools tell us? So and let us take the example of MedFrag, right? MedFrag is a really interesting tool. It was first coined already more than 10 years ago. And like Magma, it is actually has a very relatively simple approach, right? It takes structures, it breaks them, and it then matches the fragments, in silico fragments, to your experimental data. No? So, and if you think about that, then it makes sense. And sometimes it gives you a fantastic kind of suggestion, and sometimes it gives you just rubbish, right? Because that's how that tool works, right? It doesn't use a lot of expert knowledge, only in the sense that it can that it breaks the bonds at places where we think is logical. But then you feed it with all sorts of structures, it will always also present you with some relatively things that you think, that's really weird. So and I used the word common sense already a couple of times during my talk, at least twice, I think, but you have to watch back the video to count it. And I will mention it again here twice. So use your common sense there, right? If you already can see that doesn't make sense, then it's very likely that the database, for example, the structured database, didn't have the right candidates, right? Also, those tools are dependent on the candidate structures you feed they're fed with, right? So if in the database no structure is present that could explain your spectrum, then of course it will give you rubbish. Yeah. So use your common sense, and you could also try two tools and maybe see if they overlap somewhere, or as I showed you an example with the library matching, maybe you can become more confident about substructure annotation, right? And the final thing I would like to add is that nowadays, with tools like Molded Enhancer and Canopus, you could also, as a first step, do some chemical classification and then select the molecular families or groups of molecules that you are most interested in based on your question.
Yes, sir. Sir, my last question is, there are many groups, right, publishing works on, say, rapid comms of mass spec or journals of mass spectrometry, sir, detailing fragmentation of different compounds. To what extent do groups like you or other groups use this information?
So what information exactly do you mean here?
I mean, sir, there are many reports on how certain compounds will fragment that way. And right now, they currently exist on journals.
Yeah, so that's also a good question. I think we currently, most of the tools do not use that simply because it is another layer of information that you have to integrate somehow. There's a recent example in electrochemistry by a group from Seattle, Washington, and they, I think, Daniel Raftery's group, and they show how you can do molecular networking and then add on a layer of information from how flavonoids fragment. But you will notice that that's a combination then of an automated computational tool with manual kind of interpretation, right? Because they argue that you can recognize flavonoid 3-O-glycosides by the radical fragment it produces. And from my thesis time, from my PhD time, I can confirm that that's the case. But in that case, you will first do your molecular networking and then use that knowledge, apply that knowledge and some rules to then identify further, annotate your flavonoids. So, yeah.
Yes, sir. So actually, I'm very passionate about this, and I contemplated about it over the weekend. I realized that you develop MS2 LDA, right? I realized it's a good platform because it is community curated. So I mean, we can annotate certain motifs, right? But I think it will be good idea then if we can say have a tag or annotate what's within a motif. So why is it that way? Uh, Maybe some of the users have more advanced information about certain reaction. So I mean, I think that's good because it's an open platform. So that's
Yes.
all, sir. Thank you very much.
Thanks, Ralph. And let me then, let me then try to incentivize you to share back your annotations at some point in MotiveDB, the database of annotated motifs. So we want to collect more and more annotated motifs that then other people, including part of this audience, if they work on similar samples, let's say I work with plants and there's now a motif set related to plant-based motifs in negative ionization mode in MotiveDB. So if you are working with plants in negative mode, you can use that as part of your substructure analysis. And if molecules match or MS spectra match to those motifs, and they have a good match, then you don't need to do the annotation again, right? Because those motifs are already annotated. And I can tell you there's also some annotations, motif sets for urine and for some bacterial data. So, but I hope that together with people like you and others, we can grow that database. So.
Yes, sir. Certainly that's the way. So yeah, we're working and then we will publish our results. Thank you, sir.
Thank you, Ralph. Well, in keeping with your question about the Spectra published in journals, and you will be publishing soon, I just like to allude to Justin can help me with this as well. I think one of the reasons why it's difficult to use data from published papers is that they just don't report the actual raw data, or they don't upload it to a certain repository. It would be more difficult for bioinformaticians like Justin to download tables from the PDF files, right? So that's why if you are working on this, and maybe Justin can add more, you can upload your data to a repository in GNPS, of course. Maybe Justin, you can continue on how to proceed with this to make your data more open.
Yeah, so indeed, echoing Brian, it is if we want to help each other out, then it helps to not only report what you did in a manuscript or in the paper, but also in a way that it's more easily being reused, right? And there are several repositories where you can now add your mass spec data. So metabolites is one, metabolomics workbench is one, and GPS massive is one. And each of them have their own pros and cons, but let's for the example for today take GPS massive, because of course it takes you effort to get the data in, yeah, but you also get a reward, and the reward is that you can then use the whole complete GPS infrastructure. There's also the GPS dashboard where you can then make very nice pictures already, and this will further improve. You can do some statistical analysis, you can check your data, and as I said, you can run GPS molecular networking where people like Ralph already have done experience with, but and also you can share that analysis again very easily. And if the data is public, then people can even very easily reuse it as well as part of their own analysis. And another reason why you would like to do this is, I mean, who doesn't want to know whether the molecules are novel, right, or are unique, or how unique they are, or where they have been found before, so they can start to assess a function or an origin. Give you an example. If you have urine samples and you find molecules in there that are interesting, and wouldn't it be great to be able to compare them to all the public data and I see, hey, they're only found in some fungal extracts. So then you know, or you can hypothesize that those molecules are actually of fungal origin. And you know what? It's possible already, right? There's MUST. There's a blast-like tool for metabolomics. But that fully relies on data to become public and be part of the public infrastructure together with good metadata. So that is, so you see, your reward is that you can do more useful analysis, And of course, the contribution will be that you will need to spend some time on adding the data to the repositories. Yeah.
Okay, in the interest of time, I have one last question. Actually, there's one very quick question before this. What's your cosine cutoff for your identification?
So the short answer is it depends, right? You will need to use, and there comes again, your common sense to find a right balance. And one way, what you could apply is FDR, false discovery rate. That is one option. And the other option is indeed more using your common sense to assess. In some cases, 0.7 can be a good score. In some cases, it's not. And let me give you one quick reason then. The amount of noise, right? The amount of noise in your spectrum may be higher than in the library spectrum, or the other way around. And that noise contributes negatively to the score, right? So you may have a very good match for the ions of the molecule, but you also may have a lot of noise, and then your score will be still relatively low, like let's say 0.7, whereas the match is still good.
Okay. Yes. Well, just a bit of background, by the way. Ralph belongs to the Institute of Chemistry at the University of the Philippines, where they also have the mass spec facility. We have a question from the head of that laboratory, Professor Hias Junio, and this is very apt for all of us. In fact, how much programming skills do you need before you can implement these tools that you mentioned?
Okay, yeah, that's a very good question. So let me say that I would have wished that I gained more programming skills during my studies already. So if you get a chance, do it, because it will pay back itself if you continue in omics science. But the good news for you is that me and others are championing also, that algorithms are transferred into tools that can be used by, let's say, people like yourself, right? Where you have like relatively little background in programming, that you can still use the tools. I mean, most GPS tools, you don't need any coding skills to use them. Yeah? You upload the data and you have to enter parameters and it will run. Yeah. So the good news is that you don't need to put in any code. The bad news is that you still need to fill in parameters and get an idea of what they are and how they work on your data. So, but that isn't necessarily different from running a PCR and knowing how much solvent and things you need to add and with the exact temperature you can best run it, right? So I don't think, of course, it would be nice if it will become more and more easy to use the tools and know which parameters to use. But on the other hand, it is definitely worth to spend, let's say, some time in optimizing the parameters a little bit, assessing the results, and then you have a workflow that you and others in your group can use on that particular type of data, right? Because at some point you will find how you can best use the tool, and then if you run your instrument and you have similar samples on the same instrument, you can of course reuse the workflow. And I think, like I said, there's good documentation now on the internet, especially for GPS these days, and also YouTube movies that you can look at and follow. And my best advice is to really also give the examples a try, right? There's links, so you follow the link to a GPS job and explore it a bit for yourself to get an idea of what's going on. And there was the Nature Protocols paper from, I think now 1.5 years ago, that it also really nicely and in detail explains the several steps involved on, I think, 6 different platforms to get to a molecular network.
Yeah, and I guess I'd just like to remind everyone what we've been saying the past 6 webinars already. You cannot do this all by yourself. It's always a collaborative effort. So if you do have someone in your team who has more of the bioinformatics skills and somebody else has a more physiology skill and the other one biostatistics skill, it's always best that we all work together. So
Exactly.
yeah, I think Justin will agree on that.
Yeah.
In the interest of time, yes.
Note before we close is that if you decide to do that, do it early and not, when you have the data and you're suddenly stuck with doing the annotation. So try to think ahead, because there's a famous quote that many statisticians have to do a post-mortem of the experiment rather than being involved from the start and still be able to influence how best to do the experiment. No.
Yes, thank you very much. In the interest of time, if everyone has, if you have more pressing questions, you can wire it through FNRI and then we will try to answer it later on. Yes, and it is my hope, in fact, that travel restrictions permit, we can possibly invite Justin for a beachside workshop in the Philippines in the future. I hope he's up to it. Let's see. So that would be very good. Yes, so I would like to thank everyone for your participation. Before I end, I pass it back to Jacobs for some closing remarks.
Okay, thank you, Justin. Thank you very much. What you said about collaboration at the outset is very important. The lecture that you have given to us is very important adjunct to the earlier presentations, the first 5 presentations that we had. And I'm very happy how networks are slowly being built as a result of this webinar. So thank you again. And mga kababayan, baka pwede naman natin bigyan ng virtual clap our speaker, Justin, from the Wageningen University, in his very insightful presentation this afternoon. Thank you very much, Justin. We hope to see you in person once the restrictions are lifted. And as what Brian has said, we can have our beachside web presentations or hands-on lecture for that matter. All right, so I am reminding all our participants to make sure that you have accomplished your evaluation form. I think the links are now available via the chat, and perhaps our other members of the technical group would be posting the same link on our YouTube. And the certificate of participation will be available once you answer the evaluation link. And we will start giving out the certificates of participation starting October 20, 2021. So this has been day 6. Yes, Brian.
And but I mean, are we gonna remind them for tomorrow or?
Yeah, of course.
I was butting in. Sorry.
No problem. Sige.
All right. So reminder lang mga kababayan, mga kaagham, as you were saying. So tomorrow we will have a lecture by Professor Jonathan Swan from the University of Southampton on NMR-based analysis of early life nutrition. And then on the next day, Tuesday, we will have another one from Professor Lynn Van Hacke from Ghent University in Belgium about the gut microbiome diet axis using metabolomics. And again, our last lecture will be on Thursday from Dr. Liselot Hemmerich from Ghent University on DNA adaptomics. So maraming salamat po for still being with us and continuing to support this initiative and for your attention. Mula sa akin, maraming salamat po sa inyo lahat.
Salamat po. So this has been day 6 of OMICSena lecture series. See you again tomorrow. Ingat po lahat kayo. Salamat po.
Justin, thank you very much.
Thank you, Justin.
Thank you for organizing that.